Good evening. Welcome to tonight's presentation in our ongoing series, Dialogues of Discovery. I'm Jerry Rubin. I'm the Executive Director here at the Janelia Farm Research Campus of the Howard Hughes Medical Institute. Our speaker tonight is Jeremy Nathans, who's, I hope you picked up one of these when you came in, because it has a whole long biography of Jeremy. And I'm not going to read all this to you. I'm going to assume that you'll study this at your leisure. Jeremy is a professor at Johns Hopkins. He's also an investigator of the Howard Hughes Medical Institute. He's a very distinguished scientist, a member of the National Academy of Sciences, and has many other honors. Jeremy was nice enough to come over earlier today and spend an hour this afternoon talking to some of 30 or so high school students from Loudoun County in a very lively discussion. Many of you are here in the audience. I see you here, and I hope you had a good time. I want you to know when Jeremy came out and I said, well, how did it go? He said, it was great. They were really smart. When I was their age, all I was thinking about was my jump shot. So now that couldn't have been strictly true because Jeremy went on after high school to go to MIT and then went to Stanford to get his MD PhD. So he must have been thinking of something else in addition to his jump shot, although I think he has a pretty good jump shot. I don't think we'll hear about that today. What we're going to hear about is what he actually started when he was a graduate student at Stanford. So the interesting thing, one story I want to tell you about, which is only alluded to in this, is when Jeremy got to graduate school, he chose to work in a lab to do his PhD thesis research that studied how little Drosophila, little fruit fly embryos, grow up to be adult fruit flies and what happens in that process. And he was in that lab for a couple of years when he said, you know, I have this idea that I'm really interested in that I would like to work on. It has nothing to do with fruit flies. It has to do with how human color vision works. Now, you don't have to be an expert in biology to understand that understanding how human color vision works and understanding how fruit fly babies grow up are really pretty far apart. But he was fortunate to be in the laboratory of a very special person, David Hoagner, someone who was one of my own mentors. And he went to Dave and he said, you know, Dave, I think what the lab's working on is really not what I'm interested in, but I have this really great idea. I'd like to understand how human color vision works. Now, this is not normal, what a normal second-year graduate student does with their advisor, but Dave was a very smart man, and he could recognize talent, and he could recognize passion, and someone who really wanted to solve a problem, which is 90% of what it takes to be a successful scientist. And he said, sure, go for it. And now we're going to hear what happened over the next 30 or so years.
Thank you, Jerry. Well, it's a great pleasure to be here. Thank you for coming out tonight. I want to tell you about work we and others have done over many years looking at the human visual system. And this is really part of a larger quest summarized here to understand how the human brain works. I think for many of us, and I would put myself in this category, understanding how the human brain works is the great scientific challenge of this century. But it's a big challenge. It's a lot to look at all in one piece, and so one needs to break it down into smaller parts. So one way to break it down would be to study, let's say, just sensation, the different senses. But even that's a large chunk, so let's break it down further and study, let's say, just vision. Even that, though, is a big thing to study. If we ask how information is received by the eye and sent into the brain and then analyzed, there's an awful lot to study. So let's break it down even further and study just color vision. So that's the main topic of tonight's talk, and then we're going to sort of focus out on the big pictures later on in the talk. So let's ask about vision from the point of view of a physicist. What actually does vision consist of? Well, it consists of capturing photons, that is particles of light, but of a particular kind. And it turns out the kind is rather limited. If we look at the full spectrum of electromagnetic radiation from gamma rays at the most energetic onto X-rays, ultraviolet. The visible region, which we can see, just this little sliver, infrared, microwave, radio waves, and so on, we can appreciate that our visual system is extremely limited. We're just looking at this tiny sliver of the electromagnetic radiation that's out there. But despite its narrowness, and it only goes from about 400 nanometers at the violet end to about 700 nanometers at the red end, there's a lot of information there. And to appreciate that, we can compare, for example, a scene as it actually appears, the top image, in full color, with a version of it in which we've eliminated the colors. We're just looking at intensities down at the bottom. And I think you can appreciate that there's a lot more information. Of course, it's also prettier if we can see the colors. And that's the name of, information is the name of the game in this business. Organisms want to harvest information from the environment, and they use color in many different contexts. Let me just describe a few. For example, in the plant world, many plants, especially the flowering plants, are highly colored. They look pretty to us, but of course they're not doing that for us. They're doing it for the bees and the butterflies and the hummingbirds and so on, the pollinators, the animals that go from flower to flower, getting a meal, but also carrying the pollen around to fertilize the next generation. The same sort of effect is seen in looking at the ripenesses of fruits. When fruits are unripe, they tend to be green, and as they ripen, they tend to shift their colors to yellow, orange, or red. Again, this is a display that's put on for the animals. The plants are trying to coax animals into eating the fruit and spreading the seeds around. So it's clearly a form of biological communication. There are other colorations that are used to identify members of a species, for example, among insects here, each butterfly having a distinctive set of colors. In some cases, coloration is used to distinguish males and females. So for many birds, for example, the males are one color and the females are another. That's useful if you're a female and you're trying to figure out who's the optimal male to mate with. And finally, some coloration is sent as a warning signal. So if you look, for example, among frogs in the Amazon, there are many highly poisonous frogs which are brightly colored. You might think that being brightly colored, and if you're a potential meal for another animal, is not such a good idea. But the would-be predators have learned that these frogs are poisonous. And so the fact that the frog is advertising itself is essentially a warning for the predator not to eat it. Of course, the reverse strategy is to camouflage oneself and try to avoid being detected entirely. Now, let me give you a bit of an historical version of the color vision story, partly because it's beautiful and because we encounter interesting people, and also because I think there are a lot of lessons to be learned from the classic experiments. So the story begins with Isaac Newton. Here he is, arguably the greatest of all scientists. You can see from the hairstyle that he grew up in the 60s. the 1660s, I might note. Now, Newton did a remarkable experiment. It was really inspired by his boyhood experience growing up in the countryside in England. As a child, he saw many rainbows. Of course, England is a rainy place. And he was fascinated by the coloration of the rainbows. And when he was an undergraduate at Cambridge University, he decided to recreate the effect on a laboratory scale using prisms, using glass prisms. So here's the Newton experiment, the first observation. Newton drilled a small hole in the wooden shutter of his window in his dormitory room. It's not clear what his roommate thought of all this. It's actually not clear that he had a roommate. But in any case, he got away with drilling a hole in the dormitory shutter. He let in a beam of light, a narrow beam of light. Here it is. And it passed through a glass prism and was broken up into its component colors: red, orange, yellow, green, blue, and violet. For those of us of a certain era, this looks like the Dark Side of the Moon album cover from Pink Floyd. Perhaps I'm dating myself here. But Newton, just from this initial observation, made a very profound inference, and that is that white light, sunlight, is actually a mixture of all the different spectral colors. And he could put them back together again with a second prism, not shown here, essentially the reverse of this splitting up, and recreate the beam of white light. Okay, that's observation number one. The second thing that he realized is that he could use this splitting up with a prism as an objective way to purify different kinds of lights. So for example, he could build a backstop which would block out all of the rays except the orange ones shown here, which are allowed to pass through. And then he could combine those rays with others of defined refractive angle that were made with other prisms. Nowadays, we would do this with a tunable laser or a set of tunable lasers. But Newton appreciated that this was a way to objectively purify different lights. He did many experiments with these. Here, I'll just show you his pencil sketch of the setup. This is Newton's own sketch from when he was an undergraduate. Here's the window with the hole drilled in it. Here's the first prism, and here are a series of beams of light of different colors. He's got a hole right here, so one beam passes through, it goes through a second prism, and so on. And here's one of his many experiments. He combined the different spectrally pure lights in various combinations. And here, this is just done with a series of projectors. If we, for example, take spectrally pure green light and spectrally pure red light, and we shine them together on a blank surface, we get a sensation that appears identical to that seen with a spectrally pure yellow. Now let me ask, is it physically identical to a spectrally pure yellow? People are shaking their heads. Why not?
So what's happening is that the wavelength of spectrally pure yellow has a certain wavelength. What we're seeing there is the combination of two different photos with different wavelengths that just happen to look yellow to us.
Right. We've created the sensation of yellow. You're absolutely right. Now let me ask, if you're Isaac Newton and you're 20 years old and you're in your dorm room, how would you prove that that is the correct explanation?
I guess that's why I'm here.
Well, Newton had a longer time to think about it. I don't mean to put you on the spot. Here's the experiment. He took this yellow light and he passed it through another prism. And what did he get? He got the component red and green lights back again with exactly the same angles of refraction that they had at the beginning. That is, this is a superposition of spectrally pure red and green, and they have not changed at all just by being mixed. They are still their own pure red and green lights. And he realized that the angle of refraction through the prism was an objective measure of determining the properties of a beam of light independent of the sensation that it elicited. And in some cases, like this one, the sensation is not an accurate guide to the physical composition of the light. That is, the human visual system is imperfect in this respect. Now, Newton had a lot of things to say about light, and I want to share one of his comments with you. This is from his great book, Optics. He says, "May not the harmony and discord," and by that he means the similarities and differences of colors, arise from the proportions of the vibrations propagated through the fibers of the optic nerves into the brain, as the harmony and discord of sounds arise from the proportions of the vibrations of the air. This is an amazingly insightful comment. So it was known at that time that air is basically a giant three-dimensional spring. It can be compressed and it will push back, like when you fill a balloon with air. It pushes back, the balloon pushes on the air pushes back. And that sound, the propagation of sound, was essentially a series of compressions and rarefactions propagating through the air. So what Newton is suggesting by analogy is that light might also have a wave property. And this is at a time when virtually nothing was known about the physical properties of light. And that, like sound, it was propagating through the air as a wave-type disturbance. Very prescient. Let me also, before we leave Isaac Newton, just show you something about his life at that time. This is a quote from much later in his life when he was looking back on his days as an undergraduate. He says, "In November 1665, I had the direct method of fluxions." That's what we today call differential calculus, which he invented. And the next year in January, I had the theory of colors. And in May, I had entrance to the inverse method of fluxions. That's today what we call integral calculus, which he also invented. And in the same year, I began thinking of gravity extending to the moon. And from Kepler's rule, I deduced that the forces which keep the planets in their orbits must be reciprocally as the squares of their distances from the centers about which they revolve. That was a very good year. Okay, now the next big event was about a century later. And this was Thomas Young's great insight into the physiology of color vision. Young was another extraordinary mind. He was interested in everything. He was a remarkable linguist. He spoke Latin and Greek at age 4. He was a major figure in deciphering demotic text, one of the hieroglyphic texts that's on the Rosetta Stone. He was the major consultant for shipbuilding for the Admiralty in England. He was also a physician and was in command of virtually every area of medical knowledge. And one of his many interests was light, and he did a very important experiment in his mid-20s, that is, he passed light through 2 slits, and he looked at the interference pattern that results. It's kind of like dropping 2 pebbles in a pond at the same time, and he realized that this was proof that light had wave properties. So with that in mind, at age 28, he delivered the Bakerian lecture to the Royal Society in 1802, and he had this to say. This was sort of just an aside about what he thought might be going on in the retina to mediate human color vision. He says, as it is almost impossible to conceive each sensitive point of the retina to contain an infinite number of particles, and by that he means receptors, receptor molecules, each capable of vibrating in perfect unison with every possible undulation, that is, every possible wavelength. So saying you can't imagine a receptor for light of 500 nanometers and a different receptor for light of 501 nanometers and 502 nanometers and so on. There's just not enough room on the retinal surface to pack all those different receptors. He says it becomes necessary to suppose the number limited, for instance, to the 3 principal colors, red, yellow, and blue, and that each particle is capable of being put in motion more or less forcibly by undulations, that is, wavelengths, differing less or more from perfect unison. That is, he's imagining a series of broad, bell-shaped absorbance curves for these receptors with a peak at one position and less sensitivity as one gets further and further away from that position, and that there would be exactly 3 of them. This is exactly right. And about 60 or 70 years later, it was proven through a series of very clever psychophysical experiments that we do in fact have 3 receptors. Here they are shown along the wavelength axis, and here's what the lights look like down at the bottom. We have a shortwave sensor, we'll call it S, which has a broad bell-shaped curve shown here. So this is most sensitive at the peak, and it gets progressively less sensitive further away from the peak. We have a medium wave sensor, we'll call it M, and a long wave sensor, we'll call it L, and M and L are rather close together. So that's a sort of an asymmetry in the system. And these 3 receptors together mediate all the sensations of color vision. Now, can we explain the Newton color mixing experiment with this background? Yes, we can. Because a green light, as shown here, would excite, for example, the M receptor the most, the L receptor quite a bit less, and the S receptor hardly at all. And a red light, shown out here, would excite the L receptor a lot, and the M receptor just a little bit, and S essentially not at all. And a yellow in between can be created in a synthetic way by simply mixing the red and the green lights together to give the ratio of excitations that a light at the yellow position would give. Right? So that's the Newton trick. And so this neatly explains the color mixing data that Newton originally observed. In fact, there's a nice way to conceptualize it. This is from James Clerk Maxwell, another great physicist who was interested in color. You can imagine a 3-dimensional space in which one axis represents the degree of excitation of the S-sensitive receptors, another axis, the M-sensitive receptors, and the third, the L receptors. And every stimulus is some point in this 3-dimensional space. Obviously, a white light, as shown by this vector here, which is an equal mix of all the wavelengths, would be coming out of the origin, essentially coming straight out at us from the screen. Let me just leave it as a homework exercise to draw the pattern of points that correspond to the spectrally pure wavelengths. That is, just if we dial ourselves along the wavelength axis from a violet all the way out to red, it forms a particular pattern in this space. I invite you to figure it out for yourself. Now, up to this point, I've been talking about color vision as if it's one unitary thing. That is, all humans have the same kind of trichromatic, that is, 3-color vision. But that's not true. About 10% of males and about 1% of females have vision that is distinctly different from the majority. And one of the most famous people who had that sort of, or one of those kinds of variation is shown here, John Dalton, a contemporary of Young's, one of the great chemists of his time. Dalton's first paper, which he wrote when he was 26 and submitted to the Manchester Literary and Philosophical Society, was essentially an introspective paper in which he thought about his own color vision relative to those of other people. And the title is Extraordinary Facts Relating to the Vision of Colors. And here's what he had to say, among other things. That part of the image, which others call red, appears to me little more than a shade or defect of light. After that, the orange, yellow, and green seem one color, making what I should call different shades of yellow. That is, he was unable to distinguish lights at the long wave end of the spectrum, to distinguish their color attributes. What did the world look like to John Dalton? Well, here's a scene of a vegetable stand. This is how it looks to a normal trichromat. And Dalton, who we now know was missing his M pigment gene, I'll tell you in a second how we know that, would see the world this way. If you're missing your L pigment gene, you see it this way. And if you're missing your S pigment gene, you see it that way. That is, you need your long and medium wave receptors to distinguish the greens from yellows, from oranges, from reds, the long wave end of the spectrum. Now, why is that? Well, it's easy to understand if we look at the absorbance spectra. So let's just go back to the normal trichromat for a second. Here's the S pigment, this bell-shaped curve on the shortwave side. Here's the M pigment, and here's the L pigment. If you're missing your L pigment, you're down to just 2. And I think you can appreciate that all the wavelengths at the longer end are basically just being captured by the single remaining pigment. Likewise, if you're missing your M pigment, all the wavelengths are being captured by the single remaining L pigment. That pigment can only signal intensity. It cannot signal color sense. And so that gives the washed out sensation that Dalton experienced. Now, what's the basis for this? Jerry alluded to this in the introduction. This is work that I did as a graduate student. We were able to isolate the genes that encode the different light receptors, and we found something interesting. And at this point, we'll just concentrate on the long and medium wave receptors for the remainder of the talk. The two genes that encode those two proteins are side by side on the DNA. They're on the X chromosome, which accounts for the excess of color blindness among men. And they're in this sort of arrangement. It's a head-to-tail arrangement. So shown by this arrow is the gene that codes for the L pigment, the long wave sensitive pigment. And by this arrow next to it, the gene that codes for the medium wave, M pigment. The genes are quite big. So the number of DNA units, the bases of DNA from the beginning of the L pigment gene to the beginning of the M pigment gene is about 40,000 base units. But importantly, the sequences are nearly identical. So this 40,000 bases that encompasses the L gene is virtually identical, greater than 99% identical to the 40,000 that encompasses the M pigment gene. And the result is that when the chromosomes line up in the egg and the sperm and undergo recombination, there is the possibility for an error in recombination, an exchange of chromosomal material, such that additional copies are acquired or copies are lost. In fact, we got our first inkling that this was the case when we looked at a series of males who had normal color vision. I was actually one of them. I was the test case. And we found that many of them had extra pigment genes. In fact, this is my gene arrangement. It's the most common. About 50% of human X chromosomes have an extra M pigment gene. About 20% have 2 or more extras like that, the one shown down at the bottom. Now, this sort of mischief is exactly what causes the variant color vision that Dalton had. You can get a recombination event within the region between the genes, distal to the M pigment gene in one chromosome, and between the L and the M in the other chromosome. And if you just follow the recombination products, one recombination product has gained an M pigment gene. That's the one shown at the bottom. The other one, starting from here, has lost its M pigment genes, and it's here. The male who has this X chromosome is missing his M pigment genes. He's a dichromat, not a trichromat. You can also get recombination within the genes because the entire array is virtually identical in sequence. So this is maybe more interesting. So for example, a recombination event that breaks in the middle of an M pigment gene and the corresponding middle place of an L pigment gene creates two kinds of hybrids. There's the LM hybrid shown down here, and then there's the ML hybrid, the reciprocal hybrid shown over here. Now, how do we know what John Dalton had? Well, this is a fascinating story. Dalton, ever the scientist, indicated in his will that after he died, he would like for his eyeballs to be cut out and for someone to cut a little hole in the back and have a look through them to see if by chance there was a colored filter, some sort of stuff in the eye that was affecting his sense of color. That was one possible hypothesis. Well, that was done, and in fact, the view was completely clear. The filter was not anywhere to be found. So the problem was not one of filtration of the light. It was somewhere in the retina or the brain. Now we know it's in the retina. But it turns out that those eyes were preserved in desiccated form in a jar in the Manchester Philosophical and Literary Society, where they were uncovered about 180 years later by John Mullen, an enterprising scientist at Cambridge University. And Mullen got a little bit of the DNA out of those eyes and sequenced it. And it turns out that the sequence shows that Dalton was missing his M pigment gene. So he was, in fact, a dichromat, just as he noted. Okay, he would have liked that result. Let me just, to complete this anomalous trichromacy business, say that the hybrid genes encode hybrid pigments, which invariably work. The protein sequences are very similar between the normal pigments, and you can mix and match pieces of them any which way you want, and you get a protein that works. But the absorbance spectrum is shifted. So this is found in about 6% of human X chromosomes. The L pigment is now replaced by an LM hybrid, and it's shifted closer to the M pigment. Or the M pigment has been replaced by an ML hybrid, and it's shifted closer to the L pigment. And the result is that people who have that arrangement, either of those arrangements, have a smaller differential signal between those two pigments and reduced signal-to-noise ratio. There are plenty of people in this audience who are either carrying that as their only X chromosome, that is, they're males, or females who are carrying it on one of their two X chromosomes. Because, as I said, about 5% of human X chromosomes have this arrangement. Now, let's expand the discussion to our primate relatives. If you look at the natural habitats of primates, now we're not including humans because we're all over the planet at this point, but you see that primates evolved in South America, so there are monkeys in South and Central America, I should say. In Africa, sub-Saharan Africa, of course, we are African-derived, so we're in this category. Southern and Southeast Asia, and even as far as Japan. The kind of color vision that primates have comes, interestingly, in two flavors. There's the kind that we have, which is representative of all of the old world primates, the African and Asian primates. So chimps, gorillas, macaques, orangutans, us, we all have exactly the same kind of color vision. But the color vision that South American monkeys have is different. And here's an example of the sort of monkeys that live in South America. These are squirrel monkeys. And to explain their kind of color vision, I have to take a slight detour into the biology of the X chromosome. So recall that males, among mammals, males have one X chromosome and one Y chromosome. Females have two X chromosomes. Now, there's a problem with that sort of chromosomal arrangement. Females, since they have twice as many copies of the genes on the X chromosome that males have, would, you would think naively, express twice as much of the encoded proteins. And that would lead to trouble, big time. So there has to be an arrangement that normalizes the expression level of genes from a male who has just one X chromosome and females who have two X chromosomes. And nature has come up with a trick for that. It's called Xchromosome Inactivation. And it's basically a system in which during early embryonic development, when the embryo has about 1,000 cells, each cell decides, this is, I should say, a female, the trick is in females only, each female cell decides to inactivate one of the two X chromosomes, either the one from mom or the one from dad. That's diagrammed here. So here are the two chromosomes colored blue and red. They are part of the cells that have divided up to a certain point. This is really the 1,000 cell point, not the 2 cell point. And then at that point, one of the two chromosomes, in this case the blue one, is inactivated. It's still there. It just doesn't work. And in this cell, it's the other chromosome, the red one. Once that decision is made, all of the progeny of that cell maintain the decision. So all the progeny of this cell will keep the blue one inactive. All the progeny of this cell will keep the red one inactive. The result is that females, all female mammals, including all the females in this room, are mosaics. Some of their cells are expressing the X chromosomes from their father and other cells from their mother. And the cells of those different kinds are intermingled only to the extent that during development, cells have moved around and mixed with each other, which is not unlimited mixing, I should say. It's limited mixing. So, for example, when you see a cat like this one, a calico cat, some of the cells are colored, some of the cells in the skin have one color, have one pigment gene, and other cells have a different pigment gene active, and you get this sort of tortoiseshell appearance. That's an effect of X chromosome inactivation. Now, how does this relate to color vision in primates? Well, let's just look at the chromosomal basis of primate color vision. And I'm going to walk you through this slide. We're going to start with the old world primate case. That's us on the top. So here are males, here are females. So what do old world primates like humans have if you're a male and you have trichromatic color vision? Well, you have the shortwave sensitive pigment encoded by a gene that's on one of the autosomes, not the sex chromosomes. And you have the two sex chromosome located genes, the long wave gene and the medium wave gene on the X chromosome. No big deal. This person has all three pigment genes. A female has pretty much the same arrangement, but of course she has two X chromosomes, only one of which is active, but she's got the three necessary genes. She has trichromatic color vision. But the New World primates have a very different arrangement. They have only one X chromosomal visual pigment gene. And I should add that this is the basic mammalian arrangement. Dogs, cats, mice, cows, and so on have just a single X chromosome located visual pigment gene. The twist here is that in the New World primates, the gene can come in any of three different flavors. It can either be sensitive to longer wave light or medium wave light or something in between, which we've colored yellow. A male, therefore, since he has only one X chromosome, is invariably a dichromat, but he's a dichromat of any of these three kinds, depending which gene he happens to get, the red, the yellow, or the green version. But a female, if she's lucky enough to get two different versions on her two X chromosomes, let's say a red version on this chromosome, a long wave sensitive pigment on this chromosome, and a medium wave sensitive on that chromosome, and by virtue of X chromosome inactivation, which has created this mosaic of different cells, which I should add is very fine grained in the retina. So each retina will have cells of the different kinds present. She has trichromatic color vision. Now, how do we know that? How do we know that a New World monkey has trichromatic color vision? Well, you have to ask them. One does that. So this is just the schematic showing the male arrangement with the different visual pigments and the female having the two pigments. And here is such a monkey being asked the question. This is actually a holiday card from my good friend Jerry Jacobs, who's shown here. Jerry is one of the world's great primatologists, and Jerry is testing this little lady right here. She has to figure out which of these three pictures looks different from the other two. And when she guesses correctly, she pushes a bar and gets a little candy dropping down into the well here, which she loves. So we can ask, not linguistically, but by behavioral testing, whether an animal can distinguish colors. And I should just add parenthetically, to do the test right, you have to randomize intensity. And I should say, when we do it, we don't show pictures. We just show blank squares of different colors. But we randomize the intensity. So the only way they can figure it out is by color. It could be a bright red or a dim red, a bright green, a dim green, a bright yellow, a dim yellow. They have to realize that yellow is different from red, it's different from green. So we've asked the question, if New World primates have acquired their color vision just by acquiring this diversity in the gene they carry on the X chromosome, is it possible that that's the first step in the evolution of trichromatic color vision? Everyone in the field believes that probably is the case. That's probably the first step. That was sort of the proto-color vision. Ours is a bit more sophisticated since we have the additional gene duplication. Now, if that's true, then the question we went on to ask was, is it possible, therefore, to create this first step in color vision evolution in a mammal that has never had, as far as we know, color vision? And the mammal we've picked is a mouse. We can genetically engineer mice these days. And so what we did was we started with the mouse retina, which only has two different sensors, one short wavelength sensor, which is actually quite short wave. It's down in the near ultraviolet. and a medium wave sensor in the green region, and we added to their genome, we've replaced on their X chromosome their medium wave sensor with our long wave sensor, with the human long wave sensor. So this mouse is about 0.001% human. It was actually my DNA, in fact. Doesn't look very much like me, but we're on the way. Now, this is in a female mouse. So the female has one X chromosome that's a normal mouse X chromosome and makes the normal mouse medium wave sensitive protein. The other X chromosome makes the human long wave sensitive protein. The retina is a mosaic making these different kinds of receptors. The question is, can the mouse see in trichromatic color vision? And so Jerry designed a test analogous to the test he does with monkeys, except he scaled it down and made it for mice. And so what this little lady has to decide is whether this panel looks different from these two panels. And this is really what the test looks like. So the lights flash on for about a second. And then for the next second, and only for a second, a droplet of soy milk, which mice love, appears at this little tube above the one color that is different from the other two. This is all computer controlled, so we can change the locations of the colors and which colors they are and so on. And the mouse runs over, and if she got it right, she gets a little lick of the soy milk. If she went to the wrong place, it's there too briefly. And if she realizes she got to the wrong place, by the time she gets to the right place, the drop is gone. Okay, so they're highly motivated to figure out which one of these panels looks different. And here's what the test data looks like. Here, I'm going to show you some real data here. Now, what does a normal mouse do who cannot see trichromatic colors? Well, the mouse very quickly learns it's best just to guess. You just run over to one of the three and start licking. And lo and behold, you get it right a third of the time. That's the control. But a mouse that's been genetically engineered gets it right about 70 or 75% of the time. This is true as long as the test wavelength, which is shown on the horizontal axis, is sufficiently different from the control wavelength. In this case, the control was at 600 nanometers. I think you can see at these shorter wavelengths, the mice do quite well. When the two wavelengths get very close together, say 10 nanometers apart, they don't do very well at all. So these mice have acquired a chromatic discrimination capability that normal mice do not have. Now, I think this is a very provocative observation. What it means is that a single genetic change at the front end of the system that creates diversity at the receptor level can immediately be used by the brain to create a new percept. So the brain is basically, it's a learning machine. It's a plasticity machine. It's built to take advantage of variation that might be afforded to it, in this case, just by genetic accident. Now, more recently, we've gotten interested in the mosaic itself, the X chromosome mosaic. What does it actually look like? And of course, we know from the basic mechanism that there is a mosaic being created in the embryo of all mammals, but it had never been mapped out with any precision. And so we've created mice, again, genetically engineered mice, where one chromosome, the one from the mother, is one color, in this case, for example, green, and the other chromosome from the other parent, I shouldn't say the chromosome is the color. The chromosome encodes a protein, which is colored. The chromosome from the other parent encodes a different colored protein. So it's red and green in this case. And this is a retina flattened out. This is just a couple millimeters, a few millimeters across from a mouse, showing this mosaic pattern. You can see there's a patchiness. Some patches are bigger, some are smaller. And this variable patchiness has really intrigued us. Because if you look at many mice, you see that, in fact, the variation occurs on all spatial scales. It's not just nearby cells. It's not just nearby little groups of cells. Sometimes it's whole mice. So, for example, these are two sisters, same parents. This one is mostly green. She has a little bit of red here and there. This one is mostly red with a little bit of green here and there. Why are they so different? Well, it turns out that different X chromosomes are better at essentially competing for inactivation than other X chromosomes are. They're not all born equal in their competition. So even though it's random, even though it's a coin toss, it's not a fair coin. It's not always 50-50 heads and tails. In this case, it's more like 80-20 and 20-80. So this creates diversity, not just within an animal, but across the population. Now there are different females that have more or less cells from one expressing the genes of one X or another X. There's also substantial diversity on large scales within a single animal. So this is a cross-section of a mouse brain showing each cell is either red or green, but showing on one side of the brain there's nearly a 50-50 mixture, and on the other side it's quite heavily biased towards one of the two X chromosomes. We don't know if this happens in humans, but we think very likely it does, because the early embryology of humans is virtually identical to that of the mouse. And so very likely the left-right asymmetry that we have in our brains, which accounts for things like the language center being on one side of the brain, also has a random component in women. This, of course, would not be accessible to men. Men don't have the X inactivation mechanism. But among women, we would imagine there's a level of brain diversity that is distinctive that only that sex has, which may explain the different behaviors on the playground when we were in middle school. The girls are very sophisticated. You recall the boys, we would communicate mostly by hitting each other and couldn't really recall what happened 10 seconds later, but we were still best friends. So here's just another example. This is the retina I showed you earlier. That's the left retina from that mouse. Here's the right retina from the same mouse. So the two retinas are quite different from each other. Now, let's move a little bit up in the visual system beyond just the photoreceptor cells and ask how the visual information is processed. What happens after it's captured by a photoreceptor cell? Newton made an interesting observation when he was doing his color mixing experiments. I have a feeling the artists probably knew about this as well, but Newton really systematized it. He said, if you just take the lights from red to orange to yellow to green to blue to violet and put them in a natural pattern, natural in the sense that the nearest neighbors are as similar to each other as you can make them out to be, you find that it's not a linear array, as a physicist might suppose. That is, you go from shortwave to longwave, we now know, and Newton would say from least refrangible by the prism to most refrangible by the prism. You actually end up making a circle. That is, far red, I think we all appreciate this from our days with crayons in kindergarten, far red looks a lot like violet. There's sort of purple in the middle, the synthetic purple. Why should this be completed as a circle? It's a psychological construct. The answer came from the work of a fellow named Herring in the late 19th century. Herring suggested, and he was right, that the three different cone photoreceptor types are actually organized at the next level in the hierarchy in a pair of differencing operations. That is, what the retina does is it compares green middle wave cones from red long wave cones. That's one differencing operation. That's one axis. And it compares short wave cones, that's the blue, to the sum of medium and long wave cones, which is yellow. That's a different axis. So we've gone from three different cone types to two orthogonal axes, and that gives us this circular effect. Let me just show you an example of this in action. This is an after effect illusion. To get the illusion, you have to stare at this white dot. Actually, it's most effective if you use just one eye. Just cover one eye, look with the other. Stare at the white dot for about 10 seconds, and then I'm going to show you a white screen. That is all the different wavelengths in the white screen. Okay, so let's count it down. 10, 9, 8, 7. Don't let your eye wander. 6, 5, 4, 3, 2, 1. Bang. See the after image? Okay, what's going on there? Don't worry, it fades after a couple months. Well, Herring knew what was going on. What you've done in the square that was green selectively fatigued the medium sensitive cells so that when they were compared in that region of your retina to the long wave sensitive ones, this medium versus long comparison, the green ones being fatigued gave less of a signal when you saw the white light compared to the long wave ones. It's like one of those seesaws. One side of the seesaw is heavier, the other side goes up. And the same for all the other colors. Now let's go yet one step higher. And let's ask about the effect of surrounding stimuli, and in particular the brain's assessment of the illuminating light on the sensation of a given point in a scene. So I think you can appreciate these Rubik cube type objects are the same object. Here's a blue corner, here's the blue corner, here's yellow, here's red, and so on. Would you believe me if I told you that this blue that you see here and this yellow that you see here are identical. If you take a photometer and you ask how many photons are bouncing off that screen at every wavelength from those two places, they are absolutely the same. Let's prove it. We can prove it. They don't look the same because your brain has recalculated everything based on what it thinks is the illuminating light. But let's just put a little circle and then make the circle ever bigger around those objects and see what happens as we isolate them from the surrounding scene. Okay, a little circle, fatter, 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 fattest. They're just gray. They're not colored at all. Let's go backwards. I haven't changed anything except the size of the circle. Okay, let's run that play again. Got it? It's this one and that one. That is, we have decided what they really are in the real world based on the illumination. We realize this scene is illuminated with a yellow light and this one with a blue light. So we've recalculated all of the actual reflectances to give us a truer assessment of what the real scene has. But this sort of comparative analysis is everywhere in sensory perception. I can demonstrate it with an example. I need a person from the audience. Maybe you'll volunteer from the audience. Okay, step right up here. Tell me your name. Davis. Davis, so if I were to give you a $20 bill, you would be ever so slightly happier? You would be. That's good. Now, you can ask an economist why that would be so. And every economist will tell you it's not the piece of paper. I mean, the paper's pretty, but it's what you can buy with it, right? You can trade it in, you can take it to Target, you can get something for it. And so you would be, that's pretty much it for the demo. Thank you. Oh. Okay. I just wanted to prove that was true. So you would be happy to the extent that you could get stuff for your $20 bill, right? Actually, I realize I've got more of this demo to go, but stay right where you are. I'll just ask you the second part of the question. If I give you the same $20 bill, but at the same time I give everyone else in the audience $500, now are you as happy? Davis says no. But hold it. You can still go to the same Target and buy the same stuff for the $20 that you had originally, right? It's not like we've made a big enough dent in the economy that it would change the prices of anything. So since you can buy the same stuff, why aren't you equally happy? Because they have more. That is, you've done a comparative analysis, just like the brain has done here, right? And your comparative analysis has made you less happy. And therein lies much human misery. So this is everywhere in the brain. Now, let's look at another example. The white light that you see in the middle here, which looks very bright, is exactly the same intensity as the white out here in the edges. It's the same number of photons bouncing off the screen. It's just that you have used the context to decide what the white intensity should look like. Let me give you another example, this one perhaps more from experience. Here's a scene of two gentlemen in a room with zigzag walls, and you can see this gentleman is close and this one's further away. And let me just ask you, are these two blackened lines, this one here and this one here, the same size or different size? Well, there's an easy way to tell. Put your fingers up like this. Your fingers will be the ruler, and just move them back and forth between the two lines. Marku says no. They are the same. It's hard to convince yourself that they are the same. Of course, the reason it's hard to convince yourself they are the same is because if you see a scene like this, the line back here is bigger than the line up here because we're seeing the world in depth. It's further away. It's physically bigger. But even when you know that they are the same size, it's hard to tell your brain, to convince your brain that that's the case. Let me show you another example. These are images of cuneiform tablets, and they illustrate the phenomenon of depth from shading. So let me ask, is the writing here sticking out at us or going into the board away from us? Sticking out. How about over here? Is it going into the board or sticking out? It's going in. It's recessed. Now let me tell you, this photo and that photo are the same photo. I've just flipped this one around 180 degrees to make that one. Why do we think that the cuneiform here is sticking out and the cuneiform there is going in? Well, we have a hidden assumption. The sun is always above us. And so shadows that are below an object indicate that the object is coming out. Shadows within an object indicate that it's recessed. But now that you know that, try looking at this and telling yourself, well, it's really being illuminated from a light that's down on the floor. And therefore, this image has the objects recessed because the light is down below, not above. And this one's coming out at us. You can't make your brain do it. We've been so conditioned. The brain has been so conditioned by that experience that it's hard to reset the circuits. So just to bring ourselves back full circle, I think the study of vision in general, and color vision in particular, has taught a number of very general lessons about how the brain works. Some of those include, for example, how information is processed and compared, and how an internal representation of the outside world is created. We've also seen how changes, random changes during evolution, can create variation which the brain can immediately take advantage of because of its inherent plasticity. And we've also seen, personified by Dalton and the millions of other humans who have variant color vision, how variation, in this case genetic variation, creates diversity, perceptual diversity in our species. And that last point I really like, because in my view, diversity is the most precious resource that our species has. We have extraordinary genetic diversity and also experiential diversity, and we have extraordinary diversity in brain function, much of which is really unexplored. So I think this is one of the great frontiers for neuroscience for the next century, is to understand human diversity, and it'll be great fun exploring it. Thank you. Be happy to take questions. Yes.
So if you can take an extra gene and put it on that mouse retina to get the third color band. Oh. So if you can take an extra gene to put on the mouse retina to get an extra, the third color sensing, could you do that with a bird that sees beyond our color and put that on a mouse and get a fourth band or a fifth or whatever?
One would suppose now Xchromosome inactivation only gives you diversity at the level of two different kinds of cells. So you could potentially do it with a different pigment, but once you've gotten to a total of 3, you've used up the diversity that X inactivation can give you.
I guess birds have more than 3?
Yeah, so some birds have 4, for example. Chickens have 4 pigments. Now, they don't use X inactivation to create you're saying engineer a bird?
Or take a bird DNA and put it into another animal to do that.
Yeah, the trick is creating the diversity of cell types in the retina. I mean, if we could create that diversity, I think the brain would use it. We just don't know the rules at this point for other ways of creating that diversity.
Dr. Nathans, I'm Isaac. I'm a senior at the Loudoun Academy of Science, and I was just wondering, sort of on a similar note, like you demonstrated, red and green will mix to form yellow in a trichromat, but what about other animals that are multichromats, like mantis shrimp, who have on the level of a dozen receptors? How does that work? How do those receptors mix? Do they see more colors? Do they see the same colors we see differently? Are the colors where our a trichromat's probability distributions intersect at the lowest points? Are they able to see those colors better? How does that work?
Yeah, that's a really good question. I think for many organisms, the behavior and electrophysiology is lacking. So we don't have a good sense beyond the pigment content of the retina, which is information that's easier to get, what the animal does with the information. So the most wasteful approach would be if an animal simply wanted to capture a wider region of the spectrum and therefore evolved additional pigments that enlarged its capture, say, further out into the red or further into the ultraviolet. But then at the next level, they simply pooled the information without doing a differencing operation. So they wouldn't get any chromatic advantage if they threw the information away in that way. They have to do a comparison operation. I think probably the best data in that regard is in humans, where if you look at women who are carriers for one of the anomalous pigments, one of the hybrid pigments, we know they're carriers because they have, let's say, two different kinds of suns, each sun representing one of their two X chromosomes. One sun has normal color vision, say, the other sun perhaps has anomalous trichromacy. The mother must have those two different kinds of X chromosome. She has, because of X inactivation in her retina, she's got the three normal pigments, plus she has the anomalous pigment. So she is a tetrachromat in theory. And Mullen at Cambridge, John Mullen, has actually tested a number of those women, and he's found a number who will reject all of the trichromatic matches. That is, lights that are mixed together in the Newton style, which a trichromat will say look identical. She'll say, no, whatever ratio you pick, they're different. I can tell you the difference between them. So that's functional tetrachromacy. Probably a few in the audience right here.
How do we explain the creation of the sensation of color from an achromatic stimulus such as Benham's top?
Okay, Benham's top is it's a spinning top where there's black and white lines. Let's say it's sort of like a pizza. It's like a pizza pie where like one sector might be black and another sector might be white. And if you spin it quickly, you get a sensation of color that's emerged. It's because the differencing operations, the one that Herring showed, long minus medium and short minus long plus medium, in shorthand, red minus green, blue minus yellow, those operations have different time constants. So when you stimulate them with different frequencies, you bring out a differential signal between them. Those of us who work in biochemistry labs, we love that because we look at our centrifuges and you can watch them as they slow down. You get different colors spinning thanks to because the fluorescent lights are flickering at 60 hertz. And as the centrifuge spins around, it's a beautiful Benham's top.
Dr. Nathans, to be fair, I'm a clinical practitioner of vision. I'm an optometrist. And I am constantly plagued by the answer to a question which I cannot get from any visual scientists, which is when some patients are looking at a blank white screen, they perceive a greenish hue in one eye and a reddish hue in the other. It's very subtle, and nobody's been able to explain why that is. It doesn't seem to make any difference in their perception of a wide variety of subtle color variations when both eyes are used, nor does it seem to make a difference in their perception of subtle variations when one eye is used alone. They seem to be able to make these differentiations, and I do not know why that is.
I'm not sure I know why either. Is it both men and women?
Both men and women. I mean, I perceive it myself.
I mean,
I was thinking about your description of the chromosome mechanisms on left and right side with women, but I don't know why it would happen with men.
Just thinking off the top of my head, one possibility might be that one lens is more yellowed than the other. And lenses do yellow with age. Maybe they're differential in their yellow.
It does not seem to be age-related. It doesn't. I mean, I've seen it in people in their 20s and
this
people
can only be solved with the Dalton experiment, post-mortem.
It could be if we're looking. Well, it could be if we're looking at optics too. But once they're neutral in both eyes, it doesn't seem to make a difference. They still see the differential.
That's fascinating.
I haven't been able to find anybody that can explain it either.
Good evening, Dr. Nathans. I'm Kara. I'm a senior at the Academy of Science as well. I had a question about your bell curves for the wavelengths. It seemed that the medium and long wavelength curves, they had a lot of overlap, and I was wondering if there was some kind of evolutionary advantage to having so much overlap between the medium and long and having two different receptors see so many different of the same wavelengths?
That is a really good question. Of course, evolutionary questions don't generally have an absolute answer, but there is a pretty reasonable answer for that one. And that is the bending of light by a prism. When that happens in a lens, it's called chromatic aberration. It happens in our lenses and at the air-cornea interface. So if you're looking at a multicolored object, and let's say the short wavelengths are perfectly in focus, the long wavelengths will be slightly out of focus. Now, if you want to see things as sharply as possible, one strategy would be to pick a narrower spectrum of wavelengths to focus on. It turns out that the very central retina, the part with the highest acuity vision, it's wired up so that the pixel size is individual photoreceptor cells, and only there is the pixel size that tiny. Excludes the blue cones, excludes the shortwave cones. It has only long and medium wave cones. And almost certainly, by having those two spectra very close together, what the retina is doing is it's capturing as narrow a slice of the spectral domain as possible to minimize chromatic aberration, while simultaneously satisfying our desire to have a chromatic difference, which will give us the color sense that we want to have. So it's a compromise between those two competing requirements.
I love coming to these lectures, and they're always way above my head. Even the questions are. But when I was thinking about this one, and my 22-pound Maine Coon, Rocky, was sitting on my lap, I was wondering, can he see in color, and does he have the same range of color as human beings? Can cats see in color?
They're dichromats. So they only have two different kinds of photoreceptors. So we have a three-dimensional color world in the sense that all of the lights out there, if you plot them, you need 3 dimensions to plot them. Their color world can be plotted in 2 dimensions. So they have a simplified color world. Of course, from their point of view, we can't smell, right? Or hear. Yes.
So just to carry on from that question about the two bell curves very close, and your answer about the evolutionary fitness of that overlap. I don't suppose this would necessarily be in competition with that. But are they also so close because they're a mutation on the same chromosome that the longer wave receptor evolved as originally the mutation of the middle range receptor?
That's a reasonable idea. I don't think so, because in the laboratory, we've created mutations in those proteins to shift their wavelength of absorbance around. They're very tunable. So I don't think there's a chemical constraint. They could have acquired additional mutations which pushed their absorbance much further away than they now are. But they didn't. This is optimal in some sense.
I was wondering if if the pigments coded in the retina were temporary or if they were permanent, or are they constantly being coded for?
Yeah, they're constantly turning over.
Okay.
Each one lasts for about 10 days. And then it's destroyed. And so you're constantly synthesizing those proteins.
So would it be possible to temporarily to silence or deregulate one of the genes and cause temporary color blindness?
I'm not sure the answer to that. I think there would be cell biological problems with that. The cells are very unhappy if they don't have the normal amount of pigment. We and others have found humans who have mutations, especially in the gene that encodes the dim light receptor, which I didn't talk about, called rhodopsin, the one we use at night. When those cells have less than the optimal amount of protein, they degenerate.
Okay.
Yes.
I have always enjoyed the possibility of speculating to the concept that although we see a very, very tiny and select portion of the electromagnetic spectrum, that if our ability with regard to our senses in our eyes were there, that there might be a whole host of other colors out there that we have never seen. Could you speculate on that?
Well, I think every wavelength that we can't see is potentially information that we're missing. I mean, the astrophysicists have caught on to this. They look at galaxies and stars with gamma rays, X-rays, UV, radio waves, and so on. They harvest an enormous amount of information far beyond what optical telescopes could have told us.
Yes, but I was talking about color.
Oh, I see, as a differential. Yeah, I think, well, color is sort of a mental construct. It is a mental construct.
I'm sorry, I can't hear you both.
It's a comparison. So you could build a machine, for example, that would compare, 500 nanometer light to, let's say, infrared light. So we could create that synthetically, and it would give a color. Now, our brains are not built to take in more than what our retinas have given them. So I don't think we have the apparatus upstairs to work in a more than three-dimensional color world.
Hello, my name is Anoki. I'm also from the Academy of Science as a senior. I had a question about one of your displays about the retina of the mouse, and you also showed the mouse brain where you had red and green, and you said the red indicates the inactivation of a certain chromosome, and the green indicates the inactivation of another one. So what does that mean in terms of what the cell, like what is the effect on the cell? Like what is it, does it mean that particular cell sees green, or like I didn't see the link to the
Okay.
furtherness of that.
Those colored proteins were just markers for which chromosome, the mother's or the father's, was the one that was active or inactive. So it's unrelated to the red and green of color vision. But for a mouse where the two chromosomes have genes of identical sequence, there would be no effect other than just our seeing this pretty picture with the different colors. But for example, if you had, say, a human, a female who was a carrier for an X-linked disease, let's say one that affected brain function, she might show some of the symptoms of the disease depending on exactly what the pattern of X inactivation was in her brain. And that actually is well known among neurologists. So, for example, women who are carriers of X-linked mental retardation, although the males may have the full-blown mental retardation, the carrier females very often have a mild form of it, depending, and more or less mild, depending on the luck of the draw with respect to the X inactivation process.
What's up?
Last question.
So, during your presentation, you were showing how there's in some persons, there's a mutation with the L and M genome that it changed to LM and some to ML. Are we able to infer from that our gene pool will not have any pure L and pure M genes anymore? So will it be effectively a dichromat?
The devil's in the details. So some people have those hybrid genes where the hybrid is the only gene they have. They're a dichromat, but the pigment that they have is different from either L or M. So there's sort of an anomalous dichromat. Other people have the hybrid in the context of additional normal genes, and they would be anomalous trichromats. Some people have hybrids in among their extra genes, and it turns out the extras are not active. So for them, they could have normal trichromacy and not even know they have the hybrid. It's just sitting there being carried along from generation to generation.
But in the future, we wouldn't. The gene pool will not lose those pure L and M gene.
So you're thinking if at each generation we create some of these hybrids, eventually we'll sort of exhaust the supply of normal genes. Yeah, that's a good question. It's not going to happen anytime soon. But
definitely not.
It is. Yeah, I think it's heading in that direction. Let's put it that way. Now, it's all balanced by selection. So, for example, if a trichromat on average has more kids than a dichromat or anomalous trichromat, that will reset the balance. So it all depends, if you happen to walk in front of a red light when you weren't supposed to, okay, that affects the number of kids you had. Remains to be seen how it'll play out.
Okay, well, please join me in thanking you. In your folder, it tells you when the next lectures are. We actually have one in another month, which is unusual because it was canceled due to inclement weather from this original date, which was in the fall. So we have next lectures on March 26th. Date is in here.
Thank you. So you will get Doreen's crowd.